Okay. Greetings on the occasion of the International Stem Cell Day. I am Satyajit Mayor, the Director of the National Center for Biological Science, a center of the Tata Institute for Fundamental Research. We are one of the founding institutes of the Bangalore Life Science Cluster, which consists of the Institute of Stem Cell Science and Regenerative Medicine, Center for Cell and Molecular Platforms, and now more recently, the Tata Institute for Genetics and Society. NCBS's research scope is fundamental research, and it spans all scales of biology from molecules to ecosystems. And in fact, stem cells fall comfortably in the middle of this range, serving as an exciting new frontier for our researchers to engage in. But first, let me start with the basics, and that is about stem cells and its potential. Stem cells are indeed a unique attribute of multicellular life. They are found in all multicellular organisms, from plants to animals. These are cells that are set aside at the earliest stage of the development of a multicellular organism. A critical feature of stem cells is that when they divide, they generate two types of cells. One, a cell type that's distinct from itself, and another which is a replica of itself, leading to the idea and the concept of self-renewal. This capacity of self-renewal and specialization is a defining feature of all stem cells and has generated all the excitement in the field of developmental biology, opening up possibilities for regenerative medicine, something which I will touch on at the end. But let me elaborate a little more on the types of stem cells and their potential. When an embryo develops from a typically diploid, fertilized cell or a zygote, stem cells are set aside extremely early on in the process of their division and development, and these are called the embryonic stem cells. These cells give rise to a pluripotent cell fate, which result in the myriad cell types that make up a multicellular creature as we know them, or our cells. As the development of an organism processes from the early to the immature and then to the adult, more and more specialized tissues are built. And in these tissues, stem cells acquire more and more differentiated character, first from being pluripotent, in which they can form almost every cell type, to then becoming more and more, or less and less pluripotent, but also retaining the capacity to be multipotent. These types of stem cells are found in all tissues as progenitors of these cells that contribute to the tissues. They can only develop into a cell of a specific fate of a given tissue in which they are found. For example, we have mesenchymal stem cells, which are found in cells of the mesodermal layer of an organism. And a typical mesodermal tissue, for example, is a muscle. Muscle fibers are formed from myoblasts, which are continuously generated from muscle stem cells, both during the development of the muscle, and then stem cells are also deployed during the repair of injured muscle in adult stages. So they're critical, in fact, for both the formation of a tissue and for the maintenance of the tissue in adulthood. Well, a major breakthrough in the early part of this century was the discovery that one could revert the cell fate of even differentiated cells into pluripotent stem cell fates by the activation of a limited set of genes, crucially related to the control of gene expression of the so-called stemless genes. This discovery was in fact predicated, or in fact predicted, if you will, by John Gurdon, one of the founders of this field in the 1960s, when he found that the specialization of cells is indeed reversible. In really ancient, well, now ancient experiments, but classic experiments, he replaced the immature cell nucleus in a frog egg with the nucleus from a mature cell, which, for example, from the frog intestine. And he found that this modified egg cell developed into a normal tadpole. So this meant that the DNA of the mature cell still had all the information necessary to develop all cells in the frog. Then more than 40 years later, Shinya Yamanaka discovered that intact mature cells in a mouse could be reprogrammed to become immature stem cells or induced pluripotent cells that we call, comfortably iPS cells today. Surprisingly, by introducing only a few genes, he could induce mature cells to become these iPS cells. And the immature cells are able to develop into almost all types of cells in the body. This discovery, if you will, across 40 years, from Gurdon to Yamanaka, was recognized in the Nobel Prize for Medicine in 2012. I mean, very, very soon after the discovery that was made by Yamanaka. And this has indeed led to a revolution in stem cell biology and its applications. One of the great mysteries of stem cell biology today is to understand how stem cells may be reprogrammed into distinct fates. At the moment, our understanding is very rudimentary and empirical, and is based very much on trial and error. Despite this, we are able to generate a remarkable array of proto-tissues or organoids in the laboratory from either embryonic stem cells or for iPS cells. And these have already rewritten all the rules of what we are able to do with stem cells today. It's very clear that stem cells may be guided to specific fates by their extremely specialized microenvironment or the niche into which they are placed. And this niche is defined by both the chemical and the mechanical environment of the stem cell. For example, in the presence of the same chemical factors, if the mechanical environment of the cells is changed, the same mesenchymal stem cell may be driven to form cell types that are found in the bone if they are placed on a hard or a stiff surface, or cells that are found in fat tissue or adipocytes if they are present or placed in an environment that's made up of soft material. So research today is aimed at being able to parse apart the myriad clues that the cells receive from their environment, and thereby able to make, if we are able to make more defined substrates and tailored microenvironments from biocompatible material, guiding the appropriate differentiation of these iPSCs or embryonic stem cells into the right type of tissue. For example, one of the programs that we are driving at NCBS related to stem cells, along again with researchers from InStem and from the National Institutes of Mental Health and Neuroscience across the city from us in Bangalore, is to create a bioresource of stem cells from patients with psychiatric disorders. These cells are being collected from a cohort of deeply phenotyped patients who present sort of familial cases of psychiatric disorders, highly prevalent in their families. And these patients are now being followed over 7 years, and we hope over a 20-year timeline, in the hope that we will be able to understand some of the neurodevelopmental defects that accompany the diseases that these psychiatric cases carry by using stem cells from their own cells and unlock the potential for providing a unique window into what may have gone wrong in the development of the brain or the brain tissues from these patients. As one can see, this is one small part of the real potential of stem cells, which, as we all know today, can take different cell fates and help generate programmable tissues for the purposes of regenerative medicine. So with that, I'd like to conclude, and thank you all for listening to this podcast on the occasion of the Stem Cell Day. Goodbye.